I'd like to introduce Dr. Jim Karam. He's professor and chair of the Department of Biochemistry at Tulane University Health Science Center in New Orleans, Louisiana. The title of his talk will be DNA Mobile Elements and the Lateral Transfer of a DNA Polymerase Gene Across Species Barriers. Dr. Karam.
What I'm showing here is the kind of picture you would see in a standard biochemistry textbook or a review article showing how the DNA replication complex manages to make DNA over very long distances on a chromosome, in fact replicating an entire chromosome. A group of proteins referred to as the DNA replisome undergo a number of interactions and dissociations during this process, all very carefully coordinated, and all you have to imagine predetermined by evolution to be finely tuned and functioning properly. We've been studying this process in a bacterial virus called bacteriophage T4 that grows in E. coli. This is a large virus by viral standards. It's about 170,000 base pairs long DNA. It encodes about 300 proteins, including a complete set of proteins for replication of the phage genome. Although this is a bacterial virus growing in a gram-negative bacterium, the proteins made by the virus are genetically related to the replication proteins of many other organisms, including humans. In fact, the DNA polymerase of this system, called GP43 for gene product 43, is a homologue of the 3 DNA polymerases that exist that replicate eukaryotic DNA. Okay? So how could it be this a genetic relationship existing between a virus that grows in bacteria and human cells? These are the sort of issues or questions that have interested us. And we have in recent years been using what they would call a phylogenetic approach to look at natural variants of this polymerase synthesized by viruses that are related to T4. And we have looked at and in fact sequenced the total genomes of about 20 different relatives of this E. coli phage T4. These are phages that grow in Aeromonas strains, Vibrio strains, Acinetobacter strains. So that one aspect of this is that this DNA replication apparatus has actually been able to cross bacterial species barriers and function in different genetic backgrounds. This is a remarkable plastic property, you might say, of a biological system. So these viruses come in different sizes, genomes of different sizes. They share among them a certain core number of genes. That's what's been conserved in the evolution of these viruses, that have entered different genetic environments and yet are able to replicate those genomes. And we're interested in what are the agents that help these genes cross over species barriers, and what is it about this apparatus of proteins that makes it flexible enough to evolve in these different genetic environments. Okay. Now, although what I'm providing here, or what I'm saying here, deals with a biological and functional framework, these are also providing tools for us to understand protein structure and how proteins function in a dynamic fashion and are able to adapt to different situations. One of the benefits of using this kind of approach is that it provides us with the variants of interesting proteins that are more amenable to one type of analysis or the other. The enzyme, the DNA polymerase from one of those T4 relatives, a phage called RB69, turned out to be easily crystallizable. And in a collaboration with Tom Stites and Bill Konigsberg at Yale University, we were able to provide a protein and crystals that where the structure could be determined for this DNA polymerase. And it gives us the first look at the structure of this family of DNA polymerases called the PolB family. PolB family includes the replication polymerases of the eukaryotes, as I said, and also another division of life called the archaea, or archaebacteria, in addition to gram-negative bacteria. So now we have a structural framework for this protein from which we can design biochemical analyses and also interpret biological function. The protein is a multifunctional protein, carries several activities in it, including polymerase DNA synthesis activity and exonuclease activity and editing activity that allows the enzyme to correct mistakes or remove misincorporations during DNA synthesis. These are two absolutely essential activities for maintaining the stability of a genome, this allowing the constant mutation of a genome. I'm sure you've heard of HIV. HIV has a DNA polymerase that lacks the editing activity, and consequently HIV mutates rapidly, and that's how you get drug resistance. In the case of HIV, actually that's a pretty good thing because it allows the virus to evolve and adapt to new situations. Okay. For many other viruses, it's a destabilizing influence. And T4 and RB69 are an example where fidelity is maintained through the editing activity of the enzyme. So these types of enzymes are able to adapt by changing their activities through evolution to maintain stability or their survival. Now, the structure of RB69 turns out to be very similar to the structures of similar PolB enzymes coming from archaea and other sources. There have been 4 other enzymes now crystallized and examined, and the crystal structures examined. But typically, in each group of organisms, the PolB enzyme adapts certain signatures that are characteristic of that system. In the case of the archaeal enzymes, for example, which have very similar domain organization as the RB69 GP43, they have certain fingers in them that perform specific functions for that particular group of organisms. In the phage enzymes, there's typically a long, what we call fingers domain, consisting of 2 alpha helices forming the 2 fingers. And between the 2 fingers, there's an unstructured loop, a 50 to 75 amino acid sequence that is highly variable among different variants of GP43. Okay. That's a characteristic that we think is important for the interaction of 2 polymerase units, one on the leading strand of DNA synthesis and one on the lagging strand of DNA synthesis, to coordinate DNA synthesis on both sides of the DNA replication fork on it. So there are some determinants in these fingers domain for this protein-protein interaction. Okay. And then the other proteins interact with other parts of the polymerase to influence the degree of accuracy, the rate of DNA synthesis, and what's called the processivity, the ability of the enzyme to remain stuck or held on the DNA over long distances. So if we now, structural information is really available on about 5 or 6 Pol-B enzymes now, and they all reflect this type of structure. And we're trying to extrapolate from DNA sequence data of large numbers of organisms, including the set of 20 phages, okay, what has been conserved in elements of this structure all across evolution. Okay. And this is a comparison using these sequence databases of comparing Pol-B enzymes from different systems. And this is the comparison between the RKL and GP43, those that have been determined. Very similar domain structures. And based on sequence examination, no structures found. We conclude that there are certain core elements of the Pol-B structure that have been conserved throughout evolution. And then the structure modified through the addition or sometimes deletion of segments of the structure, specifically for each class of organisms. For example, one of the 3 replication polymerases in eukaryotes, Pol-alpha. Pol-alpha has DNA synthesis activity, has an active polymerase domain or palm domain, but lacks exonuclease activity, yet it has a great deal of sequence similarity to other members of this enzyme family. It lacks the exoactivity because that activity became extinct through mutation in evolution. This enzyme picked up in the process other genetic material that specifies primase, an activity that makes RNA primers for DNA synthesis and replication. Something like Pol-epsilon picked up other types of DNA that determine other interactions for this Pol-B enzyme in the replication system. So it looks like these enzymes are evolving in 2 ways, to put it in a big perspective of evolution. They're evolving by acquiring mutations, what we call vertical change, mutations that nevertheless maintain the essential activities of these enzymes to synthesize DNA and to control fidelity. And at the same time, they're evolving by what we call horizontal gene transfer, which is the acquisition or loss of big segments of DNA on this. Now, having this framework, we're interested in the processes that influence these 2 directions of protein, what we call protein redesign during evolution. And we're trying to use this group of viruses, the T4-like or T4-related viruses, as a system to look at the agents that influence this evolution. Let me explain why this is really a good system to study this sort of thing. These are some comparisons of sequence similarities between GP43s from different phages, using the T4 enzyme as a reference. Okay. Some variants are nearly identical to this protein and sequence, 95 to 100% similarity. There are some variants that are only about 30% identical in amino acid sequence, and yet they have the same structural organization and activity. So if you put this in a perspective of eukaryotic evolution. You know, if you compare mice with humans, Paul epsilon in this case, they're practically identical to each other. Mice and humans are separated by maybe 170 million years of evolution. Okay? And this polymerase, practically identical in the two species here. To see a difference of about 40, where you have about 40% identity in amino acid sequence, let's say with being a phage that grows on Aeromonas, and T4 which grows on E. coli. To see this level of difference, it's like comparing a human with a fungus like yeast. So using this group of phages, we think we're looking at a very broad spectrum of evolution of a protein. There you go. With the added advantage is that we are sure, in this case, we are certain that we are looking at genetically homologous genes. Gene 43 is conserved in all these phages. So now this is a plot up here representing the divergence in sequence, the polymorphism in sequence of GP43 over this population that we are examining. And what you can see here is that there are segments of this protein that are very highly conserved. And segments that are very poorly conserved. When we superimpose these things over the known three-dimensional structure of the protein, typically the highly mutable area, which is called mutational hotspots in here, are in joiner regions between highly conserved structural and functional modules of the enzyme. So they're susceptible to change in the flexible regions. These flexible regions are not unimportant physiologically. In fact, they are very important. Many of them are the parts responsible for protein-protein interactions. That's the dynamic part of the protein. And it is really the specificity portions of the protein that change in evolution to alter new biological functions in them. The core parts that determine essential function, like polymerase or binding the substrates for DNA synthesis, those are highly conserved in here. And it's the dynamic aspects or the dynamic portions of the protein that diverge the most. As you will notice here, there is a big drop here, a big hole, very highly mutable area. That's that 50 to 75 amino acid stretch that is at the tip of the fingertip domain of the thing. Among the phage group that we analyzed, we found several, but particularly phages that grow in Aeromonas hosts and Acinetobacter bacteria, where this region had gotten invaded by DNA insertion elements, leading to a splitting of the polymerase gene into two cistrons, two independently expressed cistrons, or at least that's what's suggested by the sequence. Okay. So in these phages are predicted to encode split forms of the DNA polymerase, meaning a polymerase consisting of two polypeptides contributing to the polymerase activity. And this slide shows a number of things that confirm that this is true for one of these phages where we have cloned the gene, expressed it in plasmids in bacteria. On the right-hand side, it shows the products from this clone, which is this band and this band, corresponding to the A and B peptides, or N-terminal and C-terminal peptides of this. And the rest of the lanes here are all controls that this is true, indeed happens. And we have purified this protein. It purifies as a tight complex of the two polypeptides. You can't separate them out unless you denature them like you do here with an SDS gel. All right. On the left-hand side here, it shows that the clone of this gene, a phage that grows in Aeromonas bacteria, is functional for supporting T4 DNA replication. This is an infection here of the clone with a T4 phage mutant lacking its own DNA polymerase. So this is a polymerase from another bacterial species, you say, or bacterial environment, that is able to do all the functional interactions it requires with all the proteins made by T4 in a totally different genetic environment. So, I mean, this is done experimentally in the lab, but the point we're demonstrating here is that it is possible that if a enzyme like this is transferred from one species to the other, there is a definite chance that it can survive in the new environment. And from that point on, it can acquire mutations and counter mutations and ultimately adapt to the new biological system. And you have a totally new biological specificity involved. Okay? Now, the implications of having a split enzyme like this, are that the A and B peptide would have to fold together in order to form the active enzyme represented here by another phage polymerase. Looking at this a little more closely, these are the polymerase catalytic and the DNTP binding sites of the protein configured. And what I'm trying to show here are that the amino acid residues that contribute to these essential activities of this replication enzyme are contributed by the two polypeptides that have become separated by these insertion elements. So that one point of this is that you can break this protein apart and you can still regenerate activity. And this is something that has occurred naturally, not just constructed in the laboratory, although we can construct the same things in just about any phage like RB69 or T4. Okay? And the point here is that these enzymes, or this class of enzymes, is so plastic and flexible that it can reform well. And that is a very important property to have for an enzyme that wants to survive evolution and mutation in evolution. All right? Now, the nature of some of these insertion elements that are able to break the genes, okay? This is one of them, something we call ICUTS, or intercistronic untranslated sequence. Somehow in the evolution of this polymerase gene, it got invaded by a DNA sequence that broke the gene apart. What you would expect to happen when there's an insertion in a gene is gene inactivation and loss from evolution. But this had to produce function immediately on insertion. And it does so because this element provided the signals to terminate translation for this part of the DNA and initiate translation for this part of the DNA. So this element must have immediately caused expression of the broken gene in order for that phage to survive. And subsequent to that, there'll be other evolution of this element. Okay. Now, there is a relative to this phage. Let me get that to back. With this information that we have on this phage and from 20 other phages sequence, we designed primers, PCR primers, and we went to the sewers in New Orleans. They're probably very similar with a tropical climate. We got raw sewage, and we screened the raw sewage for phages that are related to T4 and that can grow in the Aeromonas salmonicida strain, bacterial strain we had. Okay. You know, New Orleans, just like here, is a kind of marine environment, and they love seafood over there. So it's not surprising to find phages that grow on Aeromonas salmonicida, a bacterium that's a salmonicid pathogen. It infects salmons. But I used to think, you know, I don't do any fishing, incidentally. So I knew very little about fishing. I thought salmon came from the Northwest and the Atlantic and so on. But you know, there are a lot of, there's trout and so on, a lot of salmon-related fishes in tropical waters too. So, and the surprising thing to me, at least, was that we found huge numbers of these phages. And we were able to screen them with these PCR primers. And we found a whole population of split DNA polymerase genes that contained a variety of DNA insertion elements in them. Everything we've looked at, and we've looked about 80 phages like this, had this ICUTS element in them. So we think it's an initial event that occurred in the evolution of this gene to be invaded by this element, producing an expressible split gene. Okay. Subsequent to the entry of this element, the gene became invaded by other insertion elements. In particular, this class of elements, which we call HEG, it's a homing endonuclease gene. These are genes that produce very specific endonucleases that have the special property of creating a cut in another genome and then copying themselves into the other genome. This is called homing. You make a copy of a thing. But it doesn't just copy itself, it copies the flanking DNA with it over. Okay? So this element becomes a target for invasion by such nuclease genes through homing. Yeah? And the gene can insert here without inactivating the polymerase gene because it's outside the coding segment of the polymerase gene. There is one class of these elements that come together with an intron. An intron is a self-splicing RNA element that usually encodes a nuclease that's present inside the element. But this is a case where the nuclease is outside this element and brings in an intron with it into this element. And what I want to tell you about is yet still other insertion elements that we are finding in the called inteins, elements that splice at the protein level that can invade this region. So this is a domain in the evolution of this polymerase gene where there is a very high susceptibility to invasion by DNA insertion elements of the type that can mediate lateral transfer of this gene to other genomes. And we think that through examination of this population, we're going to ultimately be able to draw a pathway of how these elements move between different chromosomes of a population. Now, this is a closer look at the partnership of the endonuclease gene that invades the ICUTS and the intron that can enter one of the cistrons of the polymerase in this. This particular phage, or a group of phages, contain three elements in them: the ICUTS, the homing endonuclease gene, and the intron in the gene. And they're working as partners together. The ICUTS attracted the other elements in. The endonuclease, we think, is a homing enzyme for this intron. And the biochemical evidence assay really favors that model. These are results of an experiment whereby we produced the protein from this putative homing endonuclease gene. We produce it by in vitro translation and then test its activity on the number of templates coming from different phages. The DNA templates containing the sequence that ordinarily carries that intron in one of the phages. And what we find is that there are a number of intronless alleles of this gene that are cleaved by the product of this gene. So it is indeed an endonuclease, and it has specificity to the site of insertion of the intron in the intron-containing phages. This here is the site of insertion. One of these substrates here, this one, is one that contains the intron, and it's totally insensitive to this nuclease. This is 44RR. The counterpart that has the ICUTS but no intron is a very good substrate for this nuclease. Even T4 DNA in another species is a good substrate. So this homing endonuclease is potentially an agent for lateral transfer of DNA polymerase genes across bacterial species barrier. To summarize, we have identified at least 3 agents, DNA insertion elements, that are potentially agents for lateral transfer of this POL-B gene across bacterial species barriers. And we imagine that there are additional elements yet to be discovered that assist the lateral transfer of genes from bacterial, perhaps, to higher organisms in there. And these are the types of experimental systems that one could use to search and assay for such elements. How general are these insertion elements? You find the introns, well, particularly inteins, in DNA polymerase genes of archaea. And in fact, the site of insertion of the intron we find here is identical to one of those archaeal inteins. So there is a commonality, at least in target selection, between 2 sort of similar self-splicing elements. One self-splicing elements that splices at the RNA level, and the other splicing at the protein level. So although these are bacterial microbial systems, I think they have extremely broad implications to how genes evolve even in higher organisms. Thank you very much.